Hello everyone, I welcome you all once again to my series of lecture that is Understanding Pharmaceutical Science with Dr. A.J. Hariharan. So today we are going to discuss one of the isolation of pure culture techniques, that is flood plate technique. So I have divided this lecture into two parts. First we understand about the pure culture. In the second part we will elaborately discuss about how the isolation is carried out using the spread plate technique. So if you see that all the microorganisms are generally exist in mixed culture stage naturally. So in natural habitats, if you see that microorganism, whether it's a bacteria, fungi or algae, it will grows together. So in any sample, if you collect from a nature like a soil, air or water sample, you see that different species of microorganism will be present together. So this is called as the mixed culture. So similarly, if you want to study a particular organism, it is very difficult to study in the mixed culture. So a single type of microorganism, it's not able to study in this mixed cultures. That's why we have to isolate the particular microorganism into a pure culture form. Then we can study elaborately about that microorganism. So this technique was developed by the great German bacteriologist Robert Koch. So Robert Koch was developing his Koch postulates. The important thing is that he has to prove that a particular disease is caused by a particular organism. So to get a particular organism, he has developed this isolation of pure culture technique. So this technique has been widely used for exploring the microbiology in an elaborated manner to study each individual species and in-depth manner. So first we will understand what is meant by a pure culture. So the pure culture by definition and by actually it exists as that a population of cell is generally arising from a single cell. A single cell will divides continuously and it forms a colony. So this colony, this population of cell we generally regard as a pure culture of that particular microorganism. So this pure culture is needed for characterizing that particular species. So we can characterize by different means that for doing a morphological characterization or biochemical characterization or genetic characterization, we need the cell in pure form, that is in a pure culture form. So to get this pure culture, the basic ideology is that first we take the mixed cell sample, then this has been diluted into such a extent that the microbial population has been greatly reduced to a low density. Then it is placed on an agar plate so that the individual cell will be present on the agar surface that can develop into colonies after incubation. So each cell will divide and forms a colony. This can be easily visualized with on our naked eye, and it can be subcultured and used for the study. So this was the basic principle behind for isolation of the pure culture. So there are different methods available for the isolation of the pure culture. The most popular method of isolation of pure culture is one is the pour plate technique, second is the spread plate technique, and third is the stick plate technique. So in this we will elaborately discuss about the spread plate technique. So the spread plate technique is an very easiest and the straightforward method of isolation of the pure culture. So we can isolate very simply. How it is done, if you see that, so we will place a diluted sample of the microbial cell which is having a population around 30 to 300 cells. So then it is placed a single drop in the center of the agar plate, then spread with the help of a special type of glass rod called a sterile bent glass rod. Then it develops into an each colony, then it is isolated and subcultured. So this is the very easiest method of isolation of pure culture. So if you see stepwise, in the first step, we will perform the serial dilution. So the serial dilution is either done in terms of two-fold dilution or tenfold dilution, or how many folds you want to dilute it, we can do it. So I have taken an example of a tenfold dilution. So we take an original sample. From this original sample, 1 ml is transferred to a sterile test tube carrying a 9 ml of sterile distilled water. So it becomes a 1/10 to the dilution of the original sample. Then in the second stage, we will take another test tube, similarly carrying 9 ml of sterile distilled water. From this diluted sample, 1 ml is transferred. So this becomes 1 ton dilution of this diluted sample. When compared to original sample, it becomes 100 dilution, that is 10 to the power minus 2 dilution. And similarly, taken another test tube, 9 ml sterile water, then from this 10 to the power minus 2 dilution, 1 ml is transferred. So we get 10 to the power minus 3 dilution. So similarly, a serial dilution is performed until the microbial cell population has been drastically reduced. So in the second stage, we will take a sterile nutrient agar petri dish, and a small drop of sample is aseptically transferred to the center of the petri dish. Then we'll take a special type of glass rod, generally regarded as an L-shaped glass rod or a bent glass rod. Then this glass rod is sterilized by means of dipping into alcohol and showing into the flame. Then it is allowed to cool. Then it is placed over the drop. So once it is placed over the sample drop, this L-shaped glass rod forms a thin layer. While we retrieve the petri dish, what happens is this will spread to the entire petri dish. This can be done in two ways, either by keeping the L-shaped glass rod in a stagnant manner. The petri dish is placed over a rotating table. If you rotate, it will get spreaded. Otherwise, keep the petri dish stand, then rotate the glass rod. So it forms a single layer of this sample over the agar surface. So it is evenly spreaded. Then it is allowed to keep in the same straight position for 1 to 2 hours so that the microbial cells get attached with the agar surface. Then in step 4, it is the incubation is carried out. The plates are inverted and kept in an incubator at an appropriate temperature and duration. So during this period, what happens is each individual cells will divide and forms a colony. It forms a pure culture of that particular species. So these colonies can be isolated and subcultured in a fresh medium. That's why thereby these pure cultures can be studied for further characterization. So apart from this isolation of pure culture, there are different applications of this spread plate technique are available. One, we can use to study the viable count of the organism. So the viable count means the microbial organism which are living. So the living cells only has an ability to divide. So by performing the spread plate technique, what happens is the number of colonies will be equal to the number of the viable cells. So we can easily identify the colony forming units from the given sample. So the spread plate technique is the most easiest way for isolation of pure culture and to do various other activities in microbiology, especially in the pharmaceutical industries. I think this lecture will be very helpful for you understanding about the spread plate technique. So thank you very much. So continuously view my other lectures in this YouTube channel.